Alrighty, we said in the last video on amino acid catabolism that amino acids were broken down. They're basically broken down into the carbon skeleton and then into as well the ammonium group, NH4+. This ammonium group was attached to glutamate to form glutamine, and in the bloodstream, any glutamate or glutamine was transported either to the kidney, the small intestine, or the liver for processing. Most of this is going to go to the liver. Once it gets to the liver, glutamate and glutamine are going to donate their nitrogen into the mitochondria. All right, we're going to look at where this ammonium goes from the mitochondria. So while we're in the mitochondria, we have a couple different things. Bam. What we have going on in the mitochondria is we have this deposited ammonium group. All right, that deposited ammonium group is going to be combined with a bicarbonate ion, and it's going to form something called carbamoyl phosphate. By the way, as we're going through this, keep a track of where the amino groups actually come from. One of them is going to be coming from glutamate or glutamine, which has been carried to the liver from the extrahepatic tissues. All right? This enzyme here is a wonderful enzyme called carbamoyl phosphate synthase, I believe. Give me a half second to check on that. Let's actually get the urea cycle, not my other notes. All right, so this is actually called carbamoyl phosphate, and it's actually called synthetase. Why? In previous videos, we've said that the synthetase will use ATP in order to have their reactions occur. In this case, we've got two inorganic molecules that are being promoted essentially to organic molecules. In this form, they cannot act easily on the organic structures in a way that the body wants them to. In order to activate them into something that the body can use, it requires a decent amount of energy. So what happens here is we're going to have two ATP that are going to be split to form two ADP plus two PI. Sorry, just one PI. Why one PI? Because the second PI is still attached to that carbamoyl phosphate. That would form this phosphate part right there. All right. Now, as part of the urea cycle, it is a cycle. There is a beginning, there is an end. As it circles around, it's going to use a couple different intermediates. One of those intermediates is ornithine. Now, we mentioned in acetyl-CoA transport video that ornithine can penetrate the mitochondrial membrane. It can move across the membrane. So we have ornithine in the mitochondria itself. What it's going to do is it's going to bind to that carbamoyl phosphate. All right. These two ATP, the hydrolysis of those high energy bonds was used to create this bond between the nitrogen and carbon, forming carbamoyl phosphate. This high energy bond right here in the carbamoyl phosphate is going to be used to attach carbamoyl phosphate onto ornithine. What that's going to form is something called citrulline. Now really quickly, let's talk numbers of carbons for a second. Carbamoyl phosphate is a C1N1 molecule. Whoops, N1. Ornithine is a C5 molecule, and citrulline is a C6 molecule. You're always starting with C5, you're always moving to C6. Now again, as was stated in that acetyl-CoA transport mitochondrial permeability video, citrulline can also cross the plasma membrane. So once it is formed, it's going to go into the cytoplasm. Once citrulline is in the cytoplasm, it's going to be acted on in rapid succession by two things. This next step technically has an intermediate. It's going to be acted on by ATP with the release of inorganic pyrophosphate, and it's going to form citrulline AMP. This is an intermediate state that does not last for very long. This is a really high energy bond, especially this bond right there. Let's draw it in red just to make that abundantly clear. That is a very, very high energy bond. It was the splitting of 2 ATP. It's on there, it's high energy. What's gonna happen now? And this is where things start getting a little bit interesting. That AMP group is going to be switched with something else. In this case, it's going to be switched with aspartate. Aspartate, just conveniently, is an alpha keto acid of oxaloacetate. Look, acetate, aspartate. Very close. Those two are interrelated. This aspartate is going to combine with the citrulline AMP with the release of that AMP, and that's going to form a compound known as arginosuccinate. Oh man, I'm running out of room. Arginosuccinate. All right, let's do a quick recap of these carbon numbers. So we've got carbamoyl phosphate, C1N1, ornithine, C5, citrulline, C6. Aspartate is actually going to be a C4 compound. We know that oxaloacetate from the TCA cycle is C4. Aspartate, the alpha keto acid, is oxaloacetate with a nitrogen group, so it is also C4. That means that our arginosuccinate is going to be a combination of C4 and C6, which makes it a C10 compound. The next step in the cycle, and we don't need to know any of the enzymes for this, so it's kind of nice. The next step in the cycle is going to be breaking down that arginosuccinate into arginine. Now, how many carbons is arginine? Arginine is going to be a C6 compound, C6. What we're going to be breaking off of this arginosuccinate is something known as fumarate, which you may recognize is a TCA cycle intermediate, so that's going to go right off to the TCA. If you'll notice, we took out a TCA cycle intermediate in the form of the alpha keto acid, oxaloacetate. We're adding a TCA cycle intermediate back in so that we balance everything out. Again, this fumarate was C4, fumarate, however you want to say it, which means this arginine is going to be C6 because we minus C4 from C10. The last step right here, this ornithine is actually C5. I don't know why that was written there. This last step is going to break off one compound right here, which is urea. Now, urea is a little bit of an interesting compound. It's a C1N2 compound. All right? I'm going to erase this, then we're going to talk just a little bit about urea and why it's a thing. So, urea looks just a little something like this. That's not a 4, that's going to be a 2. Now, why do we want this compound and not this one? NH4 plus is a couple things. It increases the pH of the body. That means it makes it more basic. It's osmotically active due to that charged group. It's going to pull osmotically active. That charged group is going to pull water into the bloodstream based on how much ammonium there is in the bloodstream. It can be oxidized into nitrate and then into nitrite. Nitrite is in fact the DNA mutagen. So we really want to get rid of this. We don't want it in our body. What we have over here is an uncharged, not osmotically active. We can cross that off. It's not going to affect the pH because it doesn't have a large separation of charge anywhere. And this is a very stable form. It will not break down to form nitrite. Therefore, this urea can be safely excreted in the urine via the kidneys. Now, that's pretty much about it. Thank you guys for listening. Have a good one.